we go. Let's see what's going on. Yeah, of course, Catherine, thanks. When you're looking at what's actually happening here for isomerization of that retinol, what you're seeing is it's actually that's what's responsible for breaking down that cyclic GMP gated channel. We haven't taught you what a cyclic GMP gated channel is, but we did tell you is retinol is isomerized. So it's the retinol isomerization that actually closes those channels. We teach you now that glutamate release is turned off, and guess what happens? It's the lack of a signal that actually causes excitation of a bipolar cell. Okay, so this is actually something completely new. We have never seen something like that before, and that would be something I would take note of if I were you. Okay, if only because everything we've said so far has been you actually have to have a stimulus for something to occur. Well, in this case, it's in the absence of a stimulus. In this case, the absence of an inhibitory signal that actually causes excitation that allows your eyes to function. So the pathway, we'll go through this in a second. Rods and cones create graded potentials in bipolar neurons and horizontal cells. I haven't seen what those are yet, but they're going to travel through a unique pathway. And I actually spend a couple minutes describing this to you. For better or worse, we don't actually test you a whole lot on it except for how it actually gets to the thalamus. There's a specific location it goes to inside the thalamus. You want to be aware of that. Okay, so this is actually how you see in 3 dimensions, and then maybe you've had this in a different class or something like that, but you know, it's actually kind of awesome what Mother Nature is doing here. She's taking one of your eyes, so this is your left eye and this is your right eye. And what she's doing is she's splitting the optical sensory responsibilities, not only 1 in half, but 2, what she's doing is she's splitting the frame of reference. So remember, everything's turned upside down and backwards as it enters into your eye. So everything on the temporal left half of your left side of your eye is actually switched to the medial aspect of your eye. Now, was this an essay-based class? Oh yeah, I mean, I've had students write about this before, but this is multiple choice, true and false. So, you know, nudge, nudge, hint, hint, wink, wink. Make sure you understand light comes in. What's really cool here is, why does Mother Nature do that? She does it because you get to this thing that's called the optic chiasm, where she has to make a decision. Information from the medial portion of your eye is sent to the opposite side of the cortex to go to this thing that's called the lateral geniculate nucleus of the thalamus. And that's, you know, when you think about the construction of a 3-dimensional space and electrical information traveling in, you notice it actually occupies a certain area in those optic radiations. Now that left side, or the temporal side, okay, actually travels in, and notice it goes to the upper portion. When we look at what's actually happening on the opposite eye, the exact same mechanism is in place, but notice the medial portion is now it's traveling down here, occupying space would have been there, and the temporal side now travels to the uppermost portions. And this, what it does is it creates a field of view that is overlapping, and it allows now for construction of a 3-dimensional appreciation, right? So what is depth, what is height, what is width? And that's actually how you capture all that information here. Moving on to the ear. When we think about what our ear is actually doing, you know, remember, I tried to creep you guys out with ceremonious glands. You have the outer ear, which is actually the auricle and the pinna, or the pinna. And of course, it's designed to capture specific wavelengths of energy, sound in this case, and it's going to strike that tympanic membrane. It's inside your ear, and you'll see how that happens in a second. We have a series of bones inside of your ear, malleus, the incus, and the stapes, okay? And what's going to happen is, working together, they're going to move the fluid inside of this structure that's called a bony labyrinth. Okay, and it's called a bony labyrinth because literally it creates a labyrinth pathway. Okay, and there's fluid flowing inside of that. You have perilymph and endolymph, and these would be examples of liquid connective tissue. Okay, and these liquid connective tissues allow now for what's going to cause hair cells to move. Here's the cartoon of inside your figure of your text, right? So here's the auricle here. Sound travels in, wham, 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 right? Hits the tympanic membrane, MIS, malleus incus stapes or stapes, and inside the other side of this round window is a series of fluid canals. The cochlea is where we think about hearing. The semicircular canals are part of the vestibular portion. You'll notice the vestibular portion of the cranial nerve travels to those semicircular canals. When we're down here at the cochlea, you're going to see that that labyrinth I was just telling you about looks like a snail's shell. Mother Nature is going to put a series of membranous structures inside of there we'll pay attention to. Here's a more close-up aspect of, if this is the tympanic membrane, here are the three bones, and those bones are actually moving fluid on the other side of that orifice is what it's called, but it's an aperture, right? And in red here, this is what I want you to pay attention to. Here's where, if there's the oval window right there, you actually have the cochlear ducts that are creating this sort of, it literally is an ascending staircase or ascending pathway where you'll have two populations of fluids you need to be paying attention to. When we get to the vestibular piece here, notice what's happening. I mean, this is literally an arrangement of bony structures in 3 dimensions. If you've ever taken a physics class, X, Y, and Z are the dimensions, right? And what you actually have here at the base and even inside are little tiny bones that are moving, suspended, striking hair cells as a result of where they are relative to gravity. So if you're sitting up straight, the bony labyrinth that actually will be facing upwards, if this ever changes, the detection is inside the ampulla of each of these, and you'll see how that works in a few minutes. So the inner ear itself, okay, it has 3 parts to it. It has semicircular canals, the vestibule, okay? And when we're thinking about the semicircular canals, most of the work is actually being done here inside that ampulla piece. And the vestibule actually has 2 parts, the utricle and the saccule. And you're going to see what's actually moving inside each of those. I'm trying to think about what's on the final exam there, right? And it's, do you understand what each is doing? Like, what is the name of the hair cell that you find inside each of them? Okay. But let's think about how it's constructed. Remember I said this is a labyrinth. Remember I said it actually has multiple areas of fluid inside of it. And depending upon how you cut it, notice they're taking a section out of the snail's shell there, or they cut the snail's shell completely in half. Okay. And what you can see here is Mother Nature has nervous system projections traveling to unique locations, and you'll see what they are right now. What we've done is we've blown up. Let's go backwards. We've taken one small piece of this. Notice this little box right here, and inside of that, we're looking for the actual spiral ganglia that's traveling into that space, as well as the cochlear duct. And here is that spiral ganglia. So that's the nervous system piece traveling in. And on the upper part here, we have the scala vestibulae, and that has perilymph inside of it. And underneath it, you have the scala tympani, and that actually has perilymph as well. And your hearing, where you actually hear, is inside of this little rectangle. Inside of this little rectangle, there's this structure that's called the tectorial membrane. And that tectorial membrane literally is responsible for the hitting of hair cells. Here's an inner hair cell here, and there's our outer hair cells here. And the way it works is, while I'm talking right now, depending upon the vibrations that are hitting your tympanic membrane, it's moving that fluid. The fluid is now moving the endolymph inside of this space. So here's that rectangle. There's fluid sitting right on top of this tectorial membrane. And as that moves up and down, we strike those hair cells. Okay, as we strike those hair cells, that frequency, the depth, is turned into electrical signaling. I'm not going to read all of that to you. In fact, most of it's a waste of your, it's not a waste of your time. It's everything I've already told you. That bending of the hair cell actually causes a receptor potential. And that receptor potential is what's actually causing the electrical signaling. How does it work? Well, this is a sort of a summary of everything I've told you. The sound moves in, hits the bones, travels through the oval window. That oval window, okay, causes the perilymph to vibrate. That travels along, and notice what's happening is it's also causing the endolymph on the inside to vibrate as well. That's where you actually hit the hair cells. How does it actually travel once inside the brain? Well, life's a bit confusing here inside the auditory pathway, but here's what I'd like to make sure you pay attention to. The orientation pieces are, we're using the cochlear branch of the vestibular cochlear nerve, number one. Number two, we're traveling to the pons, and then we're getting input from that inferior colliculi. And that means you're refining, maybe you're moving your head towards wherever that sound comes from, and then depending upon which side you heard the sound on, you're going to now travel to the primary auditory cortex. The reason why it looks like there are different numbers of innervations here is the quality of the sound, the depth of the sound, the range of the sound, all of those things will cause slightly different stimulation. If you've ever listened to like a mashup that has different versions of a song in it, right? You'll have something from like the 70s or 80s put in with something that's made in 2020, right? When they do that, that's what actually causes all of these multiple innervations here. That's why your brain likes it. Now, the last topic before we finish off this chapter, I believe, is equilibrium. And there are two types of equilibrium. And I want you to remember back to when you were a little kid. And I don't want to hear about your stories about being a little kid. Instead, what I want you to do is I want you to think about what would happen when you went to like the playground or something like that, and you got on maybe one of those little spinny wheel things, right? And spinny wheel thing, you were holding on to it and you got spun around, and then you stepped off of the spinny wheel thing, right? And the spinny wheel thing, once you planted both of your feet on the ground, try to remember what that actually felt like. And the reason why I'm asking you is, at any given point in time while you're on the surface of the planet, Mother Nature is trying to figure out where you are on the surface of the planet. And what I mean by that is your brain needs to know where you are relative to the center of the planet. And to do this, you have two types of equilibrium. You have a static equilibrium, which is the maintenance of your body position relative to that force of gravity. Right now, maybe you're sitting, means most of the force is actually being applied to the backs of your femurs, the ischial tuberosities of your bones, but the greatest amount of force actually, of course, is traveling from the top of your skull and through each one of the vertebrae inside your body. And as your skull rests on that atlas, what's happening, of course, is your brain is like, oh, okay, I know exactly where I am. Now, if you were to get up rapidly and spin around in a circle, what's going to happen there is, one, your body's going to feel your static position has moved, and because you're spinning in a rotational acceleration, you're getting up and you're twisting, okay, and then you're beginning to slow down. We use then the dynamic equilibrium. To do this, we utilize the receptors inside the hair cells, inside the utricle, the saccule, and the semicircular canals. This is how it works. Right here, while you're trying to pay attention while I'm speaking, you may be falling asleep and your head tilts backwards and forwards. And if I called on you randomly, it might wake you up and your head might snap backwards. What's happening here is, I mentioned these now multiple times, these little tiny otoliths. These little tiny otoliths change their location because their density is slightly different. And as gravity moves, so do the otoliths. And those otoliths then drag on that otolithic membrane. And then you have these hair cells, which are made up of kinocilia and stereocilia. And that's what actually causes your brain to know which way your head is actually tilted. Okay? Number one, those are the structures you find inside the macula. When we're thinking about how it is and what it's doing, is if this is static equilibrium, this is also detecting your linear acceleration and your linear deceleration, okay? So those individuals who don't want to know, but if there's anybody out there who drives like a Subaru like WRX, something like that, right? It's four-wheel drive. You have this sort of bucket seat that's designed to hold your head and shoulders in a specific way. And when you hit that accelerator, it's designed to cradle your head as it snaps back. And as your head is snapping back, all of those stereocilia and kinocilia are being pulled back with all of those otoliths. And that's why you like the feeling of acceleration. That saccule and utricle are, of course, detecting each of these things. So we tilted our head forward, tilted our head backwards. Great. What happens when we spin? When you spin, we utilize those semicircular ducts, and the spinning is the information is collected at the ampulla. Because remember, you just got off that little spinny thing when you're on the playground, and now these have moved. But notice, if you think about it, have they moved the same way inside each of your ears? And the answer, of course, is no. So inside this ampulla, a formation that's called the cupula surrounds hair cells, and depending upon which way literally you're moving, those hair cells will be deformed. The head movement itself causes a semicircular ducts, the hair bundles will bend, a receptor potential is now detected, and that's what actually utilizes the vestibular cochlear nerve from the vestibular cochlear cranial nerve, the vestibular branch piece. So as you're, as you're spinning your head, this is what's actually happening. Now you can spin your head while your static body position is sitting still. Maybe try that. Close your eyes, spin your head side to side, right? And then open them, and then actually try to feel what it feels like. You might feel a slightly disoriented feeling, because what's happening is you're causing these hairs to spin back and forth. And while they're doing that, they're passing through that endolymph, which actually slows them down. So it takes time to get back to normal again. Now, this is a messed up pathway. And I'm going to just tell you up front, don't even bother trying to memorize it. What I want you to see, though, is how difficult it is for you to actually balance yourself in the world. So any piece that's coming from the vestibular cochlear nerve travels to multiple locations before it even thinks about ascending to the vestibular area of the cerebral cortex. Okay? And what this means is this, is that most of the processing for balance is unconscious. And you're going to notice that as you're trying to balance yourself, the input to your eyes, okay, is actually very important. Remember, a moment ago, I asked you to close your eyes. If you do exactly the same thing with your eyes open, there are some people who are very labile to motion sickness. With your eyes open, that may actually cause you to feel more nauseous. Or for some people, when they close their eyes, it makes them feel more nauseous. What's happening, of course, is for those individuals who do get motion sick on a boat, normally the treatment, of course, is to activate your body in a different way. You get one of those little wrist things that has a piece of metal in it, and you snap it into your wrist. When you snap that into your wrist, it distracts your brain from what's going on inside of your ears. But normally when you go below decks, you no longer have a visual plane to figure out where you're at. And that's what actually causes the seasickness for some. So that's chapter 17. And that means actually we have covered everything we needed to cover this semester for the text material as we get you ready for the final exam. And what I want to do is I just want to open it up to questions, because I tried to put in some review material for the final exam, as well as the review material for 15, 16, and 17. 17 obviously being the new material here. So what I'd like to do is this, and sort of just start highlighting, say inside chapter 15, what I think is important for you to know. But don't be afraid to just ask questions while I'm drawing, okay? And I'll try to make sure that it gets addressed.
I have a question.
Ryan, fire away, man. Thanks for asking something. Sitting here going crazy. Yep.
No problem. Assuming there's like a regular online quiz this week in addition to the test.
And there will be. It'll be due on Sunday, just like every other day or whatever, yeah.
And then there's no take home though this week, right?
No, it's been up.
It has?
Yeah. Go check the quiz folder. Yeah.
Oh, okay. Did you just post? I checked it yesterday. I didn't see it.
Nope. I think I put it up right after class yesterday. Okay.
Oh,
Yeah.
so it's under the test 4 quizzes folder?
Yep. Should be there. Let me get out of here and we'll check together.
Okay. And it's due tomorrow at
Oh
noon
God,
or
no.
what?
I think I made it due Sunday for you, to be quite honest
Oh,
with
okay.
you. Yeah.
I was like, let me
No.
get some more coffee. Hold on.
Yeah, no. I think I made it due Sunday, but let me check.
No, it's there.
It's there? Okay, cool. Thank you.
All right.
Cool.
Question. That was my email too, so you can ignore the email.
Okay.
Yep.
There we go. Okay, cool. I want to go back to here.
It's just I'm trying to like connect all these chapters together. Is that like a good way to, I don't know, tie it all together for the test? Or?
In fact, I think one of the questions I ask on the take-home quiz is almost exactly what I'm drawing for you right here, right? Okay. Which is, for anybody who's actually looked at it, right? It's like compare the two parts of the autonomic nervous system. So, you know, this is like a great question to get you ready for the test, in the sense that, remembering that if up here is the sympathetic portion and down here is the parasympathetic portion, right? You know, a compare and contrast question is one of those ideal questions that helps you sort of get granular pieces of data and put it into the useful, the useful places. And some of the simpler things I would expect at this point in time everybody to be able to do without even thinking about it is like, all of the motor outputs of the autonomic nervous system have two parts: a pre-ganglionic neuron and a post-ganglionic neuron. The moment you do that, it helps your brain go, okay, I know I need two types of neurons I need to have the knowledge of. And because we know that all of our preganglionic neurons exocytose acetylcholine, it means now everybody inside the class knows that every single preganglionic neuron is a cholinergic neuron. Okay? The neuron that exocytoses acetylcholine is a cholinergic neuron. And this is when somebody pauses and goes, but you just told me it's a preganglionic neuron. Well, you can have multiple names. Each of those names describes a different responsibility, right? Now, that postganglionic neuron for the sympathetic nervous system, though, now it releases norepinephrine. I'll just put NE here, right? And for chapter 15, 16, and 17, you know, all we care about is that it releases norepinephrine. We don't care what norepinephrine is, but inside chapter 15, we teach you that this now makes this an adrenergic neuron. Adre. And notice it's unmyelinated, so, you know, for the final exam, we'd expect you to know that that's continuous conduction, but for this class, it's pre and post. It's an adrenergic neuron, okay? Down here, our postganglionic neuron exocytoses acetylcholine, ACH. And therefore, that makes the postganglionic parasympathetic neuron a cholinergic neuron as well. And that's that for me, you know, thinking of way back when I was learning all this stuff, that is where most of myself and my classmates would have difficulty. And I've tried to figure out a way to distill. I mean, this is essentially a distillation of the entire chapter for you in one cartoon. What it doesn't cover is this type of information, meaning that one cartoon doesn't cover this type of information. So let's draw this for you. I think I drew something like this for you yesterday, right? Ball and stick. And then what I did is I changed colors for you. 1, 2, 3, 4. Notice I'm telling you the number, and it means that means something special to you. There are 4 cranial nerves that are responsible for the cranial, pelvic splanchic portion, cranial sacral portions here make up the parasympathetic nervous system. So I'll go like this for you. Para. And now, yesterday I did this for you. Remember, I'm not going to draw out all of the blue lines, but I want to make sure you see what's happening. Those blue lines are coming out from a specific area in the spinal cord. And yes, you need to know that that is pretty much the thoracic portion of the body, beginning the lumbar portion, but it creates this structure that we call the sympathetic trunk ganglia. And this creates problems for people because there are some ganglia that are inside of it. Uh-oh, shit's happening on my side here. Sorry about that. It says my internet connection is not stable. If it fails, I'm just going to continue to record this and I'll place it up on Blackboard for you. Now, how do you use this cartoon? Because, great, we've broken up cranial sacral portion. We have now the thoracic and lumbar locations where the cell bodies exit from the lateral gray horns to now travel into the sympathetic trunk ganglia. And this is where you need to parse some of this information up here. What I mean by that is, in some instances, especially down here, you'll have a solid line that exits, and that still means that that is your B-type fiber. Okay, so that's your preganglionic neuron if it's solid because it's myelinated, and then it will form some form of a ganglia outside of the spinal cord. And yesterday I pressed people to go look this up to make sure that you got it right, and you remember that these were prevertebral, right? Prevertebral. And from my perspective, at least remembering back to when I was in your place, okay, it was easier for me to remember what the terminal ones were because I wanted to learn what the cranial nerves were. So terminal is here. So terminal for the parasympathetic were the ones that I learned. And that means, if you're thinking about it, if you're trying to maximize neuron utility, right? If you learn these and you learn the cranial nerves, it means the likelihood you're going to identify a sympathetic prevertebral ganglial structure as a terminal one is lessened, right? And that's where people make mistakes. So you want to make sure, yes, you're aware of where these are. And I want to point out one thing to you, and that is, hopefully you got this from the reading, but maybe you didn't. You'll notice that the vagus nerve, one isn't part of that, and it actually creates its synapses where it activates its postganglionic neuron at the target tissue. Okay, so this is actually part of the reason why it establishes and maintains normal homeostasis. So literally the postganglionic neuron that activates here the sinoatrial node inside of your heart, it's actually the unmyelinated cholinergic neuron that's releasing acetylcholine onto that part of the heart. And you'll notice today, what I did is I added just a little bit more refinement to this cartoon. Okay, but the idea, of course, is getting back to what we drew yesterday. Let's see, like this here. Maybe put some smooth muscle down here for you. Now what you can do is you can just draw inside your mind's eye. I mean, obviously you can draw it on the test, get a sheet of paper, do whatever you need to do, short of, willfully cheating. But you can see that you can release now epinephrine and norepinephrine here, but there might be some parasympathetic antagonistic innervation. It's going to release acetylcholine here as well. And this is actually how it works, and you can do that for each one of these. But that ultimately is chapter 15 in a nutshell. Okay.
So, I have a quick question.
Yeah, fire away, man.
So why are there two sympathetic preganglionic neurons in your cartoon?
So, no, you caught it. Remember, this is one of those exceptions. Remember this here, this really bad cartoon that I've drawn for you here. This, let me make sure we're in the right color here. Sorry about that. Okay. This here is the adrenal gland. And that adrenal gland within it has the adrenal medulla. And inside the adrenal medulla is a population of cells that are called chromaffin cells. And those chromaffin cells, when activated by acetylcholine using nicotinic receptors, release norepinephrine and epinephrine into your bloodstream. So your sympathetic nervous system can go directly to a gland and then cause that release directly into your circulatory system. And that's where you would have, say, a global effect of epinephrine. Did that answer your question?
Yeah, but is it always being released simultaneously to the one above it, or you're just showing the two different possible outcomes of a preganglionic neuron?
Yeah, I'm showing you the two possible outcomes. What I was going to do is ask you, hey, have you ever been in a car accident before?
Yeah, but never a severe one.
Right, okay, but you remember that feeling of, your hands shaking afterwards, or your stomach feeling sick to its core? Yes,
Yeah.
no, maybe?
Yes.
Okay, fantastic. That feeling you had enough of a sympathetic response that caused a global release of that epinephrine and norepinephrine. But normally every moment of your life is regulated by this sort of lower level preganglionic, postganglionic antagonism with the parasympathetic nervous system. Okay?
Okay, thank you.
Yeah, so it's only under special circumstances.
Okay.
To add on that,
Sure.
so yeah, I was on the highway, it was like a year ago, and I almost hit a car in front of me because like everyone in front of me like slammed on the brakes.
Mm-hmm.
You know how that happens with like traffic, and I didn't see it.
Yeah.
Luckily, it didn't hit anyone, but man, I was like wired after that. Like the adrenaline was coursing.
Yeah, and essentially what that was, if you want to put the whole story together, right? Remember, your superior colliculi saw something, it caused a global autonomic response, which actually caused the sympathetic preganglionic neuron to mass activate both adrenal medulla portions to release all the epinephrine and norepinephrine inside of your body. And this is how you tie the loop to make sure you understand it. Why were you amped? You were amped because every single receptor inside of your body that can be found on any given cell, both excitatory and inhibitory, right? So all alpha 1s, beta 1s, alpha 2s, beta 2s that bound to that neurotransmitter were now activated. And that's why you felt as hypervigilant and possibly as nauseous as you did. The nauseous piece is literally, it stops blood flow to your digestive system. And that's why you feel nauseous. Okay? Glad you lived. I'm glad you didn't kill anybody.
Yes, absolutely.
It happens. So
yeah.
Okay, so that's chapter 15, right? And notice what I did there. I mean, I can essentially put, and hopefully by the time you sit down to take the test, maybe you don't draw everything I drew here, but you have enough of some sort of frame of reference so you can think through any question. And once upon a time, back when we were used to be on a campus and stuff like that, I would tell people that I keep people's exams, right? So at least once upon a time we had exams you could actually touch, right? And you'd see that the students who did really well on the exams were the ones maybe didn't draw exactly what I drew here, but they figured out some mechanism to keep the systems separate from one another, okay? So because it's really easy to confuse 3 different preganglionic neurons here, but if you know what the targets are going to be, glands, cardiac muscle tissue, and smooth muscle tissue here. Notice I'm not stressing which receptors you find on each of these. I'm stressing that it's antagonistic because it could be activated by parasympathetic or sympathetic stimuli. So let's get rid of that. Let's do this. Let's go. New share. Let's do this. Okay. Let's get you out of the way. Let's get you out of the way. What do you guys see on your screen right now? Could somebody tell me, please?
Blank white.
Thanks. Cool. So I drew those for you. Let's compare. Let's do this. It's supposed to say ACH, right? blue over here. Okay, so here's the basic wiring diagram for chapter 16, right? Where you have some set of receptors that will send some information to an unmyelinated neuron that will synapse at the thalamus, okay, to then synapse at the brain, at our post-central gyrus. So let me write that up in blue, because it is in blue, right? So let's go post. And central gyrus is CG. Now that means that any motor output as a result of that is going to cause some electrical event at a pre-central gyrus, CG, which will then send an upper motor neuron scurrying around the brain, sending information down to a lower motor neuron. Okay, and then the real basics are where students tend to fall apart on these types of questions. If you're a lower motor neuron, how are you exiting the spinal cord? Anybody? You're using the ventral horns, right? So you're using the anterior sets of horns will then just come out, travel through a nerve, depending upon where they're going. And you notice we didn't really stress where this was happening. We just said that it was happening. Instead, inside chapter 16, what we do is we focus on what are the major muscle groups you might have to move. Is it moving something close to the trunk, or is it moving something at the distal ends of the trunk? Okay. And now because of that, what this means is when you're studying it, this is what I'm hoping is going to be a useful tool for you, is if you break up, if you spend your time literally before you start trying to memorize, you know, the pathways. The cartoon's easy enough to draw. Look, it's 5 lines. 1, 2, 3, 4, 5. 5 lines. The cartoon is easy. If you can spend some time thinking to yourself what type of stimulus is being detected, stim. Notice how I tried to group it together in class. Is it something that keeps you safe? Is it something that's just purely informational? Is it something that's required for the front of your head and the back of your head, right? Those are the 3 major groups, right? And if you can figure that out, then you can figure out which one of these pathways you'd be using. Likewise, for motor output on the pyramidal pathways, we were interested in thinking about what moves muscle groups that are close to the trunk versus muscle groups that are distal, farther away from the trunk. And if you can do that, then you have a really easy way to sort of group these pieces of information together. So you're always asking yourself, is it close to the body? Is it farther away from the body? And then you think about which pathway would be used. So that's the first thing, number 1. Number 2, the other piece of information that seems to give students difficulty, okay, so if this is trying to help you categorize the information, either ascending or descending, it's how do you describe the information? And by that, I mean, unfortunately, you have to spend some time looking at things that we would call, say, exteroreceptors, extero, and intero. And just speaking for myself, right? Things like detection of blood pressure, things like detection of your hunger state, all of those easily thought of as interoreceptors, right? No ifs, ands, or buts about that. Where people get confused is what's an exteroreceptor? If I'm you, I'm thinking all of your dermal receptors are external receptors, right? Your ear, your eyes. And in fact, what I'd recommend is when you start, I mean, we just finished 17, right? So there's no way you want to sit down and read that again. But when you're studying for the exam or you're writing, you know, a write-up for, I think I asked you about the ear on your take-home exam. I switched it this year. Okay, so, but, you know, when you're thinking about it, think to yourself, oh, this is an interoreceptor. This is an exteroreceptor. Because that modality, sorry about this, guys. That modality is what's going to be important. Now, if you're a bag, so last night we finished up everything inside AMP2 early. We're done, right? So we went through why is it humans are a bag of salt water. And well, you're a bag of salt water, you know, like, not that you really want to worry about this, but it's all you are, right? And this bag of salt water has to have certain amounts of pressure inside of it. So types of pressures that you may not even think are important, but are actually really important, are things like osmoreceptors. You know, the osmolarity inside of your body is osmolarity. And part of this is getting you ready for next semester, right? Making sure you understand that once all of these receptors are in place, and the major osmoreceptors are actually inside your hypothalamus, okay, which makes sense when we think way back to the beginning of AMP1 when we said, hey, homeostasis is regulated from your hypothalamus. What this means, of course, is that if you understand what osmolarity is, it means you need to put interoreceptors in locations where this can be detected. So osmoreceptors, baroreceptors, chemoreceptors, these are all inside your body, and these are all collectively interoreceptors. I think people get freaked out about this stuff inside chapter 16, okay, and they forget about this stuff, and you don't want to forget about this stuff. Okay. The last thing I would probably add to this is inside chapter 16, remember what we did is we switched away from graded potentials to receptor potentials, and generator potentials. And there's that one figure, generator potentials. There's that one figure that says, look, pretty much if we're using a sensory unipolar neuron, which type of potential is collected? Is it going to be a receptor potential? Is it going to be a generator potential? Why aren't we calling it this anymore? In fact, we don't even get into the why if you pay attention to it. We now just say inside chapter 16, we've moved away from a multipolar neuron. We've moved now to a unipolar neuron, and the unipolar neuron uses this type of potential. I think that would be most helpful for you. Okay. Let's erase all of that. Do I have any questions about this stuff here? No? Okay, so let's get rid of that. Clear my neurons. Let's go to this. You are a bag of salt water. It has a series of structural modifications that collects information, travels into and out of your brain. And if we think about the subset of special senses, right? The specials inside of your body, if you think about one way to use this, okay, to review for your final exam, but I mean, even then, I mean, the same questions are generally topic-wise pretty much the same. If you go from the cranial nerves, which cranial nerves make up your special senses? If you have 5 senses, right? Which cranial nerves did you use to, 1, detect? And you'll notice DETEC. That's what we covered inside chapter 17. We didn't talk about the motor outputs so much. We talked about what was the pathway into the central nervous system. And notice the pathway we spent the most time on was your visual system. In fact, in lab, I mean, essentially all we focus on inside labs, I'll show you a tongue, great, whatever. I'll show you, yeah, we won't even cover balance inside lab. All we do is pretty much take apart an eye and take apart an ear, and we want to make sure you spend most of your time thinking about those. Doesn't mean the others aren't as important. What I'm saying is that the pathways for those, they have some oddities, and maybe you want to pay attention to that. If you think about where I started lecture today, that lecture started with, hey, when you blink, this happens. When you open your eyelids, this happens. I do want to point out that I did show you this structure that was called the lateral geniculate of the thalamus. And it's the only time we have ever given you that much detail when we're sending sensory input in. when you compare it to, say, the olfactory system, which does not travel to the thalamus at all, there's something interesting going on there. And I'm not expecting you guys to be able to tell me which side of the eye goes to which optic radiations in the opposite hemisphere or same hemisphere. I am hoping, though, that you realize that the eye is split in half, depending upon its responsibility. Not quite sure what else to say about chapter 17. If I'm you, you'll notice I wrote out each of the pathways for you. Don't try to commit them to memory. Try to make sure that you'll see I told you which type of potential was used in them, though, okay? So switching between a receptor potential or a generator potential, you want to pay attention to the ones that I pointed out to you. I'm not quite sure what else to say in terms of development of that information. Each of the special senses, essentially it's going to be, look, what's smelling? Remember, that is a chemical odorant, binds to a receptor on a cilia. Okay, vision is photons striking photoreceptors. I don't care if you remember the frequency of wavelengths is 400 nanometers to 900 nanometers. That's not useful information. What is useful is what's the difference between a rod and a cone? Red, green, blue. What does that actually mean? And by that, I mean, if you're red, green, blue, are you using a rod or are you using a cone? That's the sort of stuff you want to make sure you do not confuse. The physical structure of the eye, there are chambers with fluid. And remember, yesterday I spent most of my time discussing the hyaluronic acid and what that actually does to maintaining the shape of your eyeball. Okay? What this actually means for you is, I'm probably going to ask questions about hyaluronic acid. I'll probably ask questions about the anterior chamber. Why does Mother Nature put those fluids there? She puts those fluids there so that light has a specific distance to travel inside of your eyes. And anybody who's wearing glasses or contact lenses, all those do is change the distance and the angle of the light before it hits your lens, so you can resolve that information. Let's see, taste, 2 seconds, right? You have hair cells that have 5 different types of tastants. Beyond that, though, the 3 cranial nerves. That was the important part, right? How do you actually detect the different pieces of information? Let's see, hearing and balance. Balance is going to be too hard to actually ask questions about, except for where is it detected? It's detected in the semicircular canals and the ampulla, okay? You have 2 types of balance. You have static and you have dynamic balance. Versus hearing, which is just using those hair cells that have been activated by the tectorial membrane. Once upon a time in my previous life or a previous life, we would take apart animals to look at the specific hair cells so you could actually measure the force that was required to activate those hair cells and then try to generate mutations to either increase or decrease their acuity of hearing, right? And the idea, of course, is at the time, people didn't understand how it was the tectorial membrane actually hit those hair cells. So even though that was like a gajillion years ago, that really basic idea of force activating a mechanoreceptor to allow an ion in, that's an important concept. It's a repeated concept and similar to what we taught you way back inside chapter 12. Okay, don't know what else to add there, but I'm open to any questions you have. So if you have them, don't be afraid to fire away. I will try to get the multiple choice, type practice quiz thingy up for you guys. Yes, you get 2 stabs at it, select the higher of the 2 grades, and definitely helping you to focus on materials for the exam. And then,
The Sunday exam, is that going to be open until midnight again?
I will most certainly make that available to you till midnight, yes. Woohoo! Oh, speaking of midnight, I will also be posting, for those people who are coming in on Monday and Tuesday, right? I'll be posting on, they haven't told us, what you guys are going to experience when you walk in the door, short of make sure you show up with your own goddamn pen so you can sign in, okay? I'll send you an email message sort of telling you which campus to go to, what I think you should be doing. Like, it's a lab. I mean, the building's air conditioned and all that sort of stuff, but you can't show up with open-toed shoes. You can't show up in shorts, that sort of thing. So you'll get a piece of information regarding that. But beyond that, I will have put together a sort of, this is what we're going to do video. And you don't have to watch it before you come to lab, but you want to watch it probably after you've come to lab, because I'll have you do a series of things. But of those series of things, there's going to be a subset that I want you to pay attention to get you ready for the practicals. Okay. Beyond that, I guess have a wonderful, dreary, gray day.
Wait, you said Monday.
Good, Ryan.
Monday.
Yeah, good.
Monday, Wednesday, right? And then Tuesday, Thursday, not Monday, Tuesday.
Yeah, but so it's this. They're two different groups of people, but their first time coming to campus will be Monday or Tuesday, right?
Okay, yeah, I got
yeah,
it.
But you're right. So if you're the Monday, Wednesday group, you'll get that information on Monday. Tuesday, Thursday group, you'll get that information on Tuesday, sort of stuff. So you actually have peace and quiet time now, right? You don't have to be online at 8 o'clock in the morning to listen to me talk. And as you asked yesterday, there's no lecture time. It's not like I'm doing multiple things at the same time. When I'm in lab, I'm thinking about lab.
Okay.
Yeah. So, if you're a Tuesday student, that means you've got Monday morning free, but make sure you use that time properly, right? That makes sense. And I would use that time.
By 8 a.m.?
I'm sorry?
We have to be there by 8 a.m. though, right? Whatever our day is.
Yeah, I think that's going to change as we get into like the second week and I figure out who can do what. I just don't know who has what skills yet, right? So yeah, I'd suggest you be there by 8 so I can figure out, what people can actually do. And who knows, maybe we'll even change that by Wednesday. Okay.
All right, and you'll send us an email with the building?
Yep.
All right.
Okay.
I have a question. Do we need to bring any notebooks, or how is all the data that we collect going to be recorded?
I would I'd recommend bringing your cell phone. Usually I'm not a person who suggests to people to take pictures of things, but I have to give you a practical that's essentially going to be online. So working from images, so if I point out to you a specific model, and I want you to pay attention to a specific region of that model, you're right. I don't want you bringing in a lab book. I don't want you doing all that sort of stuff. I don't want this to become problematic. But you're carrying around your cell phone with you all the time, right? So I would bring that, and you're going to be spending a lot of time just manipulating things. It's expected everybody's going to be wearing their mask. You'll be wearing gloves, so there should be relatively little likelihood of problems between sharing models and things like that. But the thing you can't do is you can't do dissections as a team, which is how we would normally do them. You'll be doing your own dissections. So make sure, you take a picture of the dissection you do, okay? Does that help, Nicole?
Yeah, thank you.
Sure, no problem. And obviously I'll be writing everything out on the board ahead of time, right? So you can, at that point in time, take a picture of that and then sort of compare it to what I actually want you to pay attention to. Make sense?
Yeah. Mm-hmm.
Okay, cool.
I'll make sure to get my hazmat suit.
It's kind of funny. Long before they ever did that. That's actually the way they approach it in the microbiology course here at the college. I'd swear you're like in a BL level 4 sort of like Ebola virus infection state in a negative pressure room. But as long as you keep your mask on, as long as if you leave the room, you wash your hands, all that sort of stuff, you should be fine. I mean, we have a pretty good idea now how everything spreads. We're inside a room with reverse pressure. Everything should go out through the ducts. Barring somebody doing something really foolish, everybody should be, I would say, above moderately safe. How does that sound?
That sounds good. I'm not too worried about it anyway.
Cool. Good. So, I mean, if you have questions about the take-home, if you have questions about the quiz that I'm going to put up, don't be afraid to shoot me an email message. Otherwise, test will go up on Sunday, or I'll go live on Sunday. And as Caleb asked, the test will be ready to stay up until midnight. Okay.
You were about to say how you think the Tuesday group should utilize the time on Monday. What were you about to say?
I'd get ready for your final exam. Yeah. So
That's,
you know
that's
that.
where I thought you were going, but
Yeah, exactly right. I mean, there's another section that's being taught at this point in time, and the instructor was adamant that they didn't want to do any lab work until they had taken the final exam. And I'm like, well, what's the point of that? You can use the lab material to review the material on your final. And this person just didn't want to be bothered, so he's making them take their final today. I would suggest you use this time to review what you're doing, right, in lab, and compare and contrast it against what the topics are for the final.
Okay, one more question.
Sure, fire away, Nick.
Do we have to do like, let's say, like a post lab, like online?
No, this is really about making sure you have the experience of cutting through all of the different dissections we were supposed to do. If I'm you, the way that I've had students successfully do this in the past, something along the lines of they create a montage of, you know, the structures we want to make sure you cover. And the way to think about it is like, he's going to think about me, think about the fact I'm going to take what's the most important piece of information here. How am I going to give it back to you in terms of an image that will have you then identify? If we use, say, an example for this upcoming week, can you identify the nucleus of a cell? Well, everybody inside this class can do that, right? But can you find the centrioles inside of a cell? That's a little bit different. So you want to pay attention to those differences. Does that help?
Yeah, okay. I just wanted to know if you were like actually assigning like any, let's say, like quote unquote homework for lab, but no, it's just kind of like whatever we do in lab, that's just like all the information that we need. If we want to put it into like our own notes and organize it, then that's what we can do. But okay, I get it now. Thank
Exactly,
you.
right? You're going to see that one of the things I'm going to send you, and I think it's already up on Blackboard already, is we have a list of structures that I'm using that open educational resource lab book for to find. So like when I put something together for you, you know, a little 15-minute video to say, so today we covered this inside lab, you might want to pay attention to this, this, and this. You know, you're going to compare that to what you saw inside lab, so you're ready for a practical. Make sense?
Yes, gotcha. Thank you.
Of course. Yay. Well, I hope you all have a wonderful day. And, you know, think about that. We've covered 17 chapters of material, and you should be pretty impressed with yourselves. Because, I mean, I think it's still going fairly well. So, and one way to think about lab is this will help you reinforce those topics that we know we're going to see on the final exam. Obviously, I'm not going to make these practicals impossible things for you. You're not putting your hands inside boxes. You're not doing all of the normal things that I think would make a lab a little bit more uncomfortable. This is more about having the experience of making sure you did things to make sure you understand how they're built. Okay? So have a wonderful day. I'll put this up on Blackboard as soon as I, you know, convert it and send it up onto YouTube. Okay? So be safe. And I'll see some of you on Monday, and I'll see some of you on Tuesday. Take care now.
All right. See you later, Prof.
Hey, take care, man.
Thanks.